Protein synthesis is the fundamental, biological process by which cells build proteins. This process follows the central dogma of molecular biology. The central dogma represents the flow of genetic information. First, DNA, which is the master blueprint. Second, RNA, the messenger that carries instructions. And third, protein, the functional molecules. This involves two steps. Transcription, where DNA becomes mRNA in the nucleus, and translation, where mRNA becomes protein at the ribosome in the cytoplasm. Picture 1. Simplified central dogma diagram. Let us look at the visual breakdown of the central dogma. On the left side, we have the DNA molecule. It is a double helix with a sugar phosphate backbone and nitrogenous bases like adenine, thymine, guanine, and cytosine. In the center, the arrow represents transcription. This is the synthesis of RNA from a DNA template. This creates the mRNA molecule shown in the middle. Unlike DNA, it is single-stranded and contains uracil instead of thymine. It carries the genetic message from the nucleus to the ribosome. The next arrow represents translation. This is the decoding of the mRNA sequence into an amino acid sequence. On the right side, we see the final protein. It is a folded 3D polypeptide chain made of alpha helices and beta sheets. These functional proteins are now ready for biological activity. This picture 2 shows cellular-level central dogma. Now let us look at the process inside the cell. The cell is divided into two compartments. On the left is the nucleus, which houses DNA and is the site of transcription. On the right is the cytoplasm, where translation occurs. Transcription process. Inside the nucleus, the DNA double helix unwinds. The enzyme RNA polymerase, shown in orange, binds to the DNA. It moves along the template strand to synthesize a new mRNA strand. The base pairing ensures accuracy where adenine pairs with uracil and guanine pairs with cytosine. Once complete, the mRNA exits through the nuclear pores. Translation process. In the cytoplasm, the ribosome, which is a purple and green structure, binds to the mRNA. The ribosome has three sites called the A site, P site, and E site. Green cloverleaf structures called tRNA bring specific amino acids to the ribosome. Each tRNA has an anticodon that matches the mRNA codon. As the ribosome moves, along the strand, a polypeptide chain of red and purple beads begins to grow. This chain will eventually fold into a functional protein. Summary and key learning points. To summarize, the information flow is unidirectional from DNA to protein. It is a two-step process starting with transcription in the nucleus, followed by translation in the cytoplasm. Molecular machinery like RNA polymerase and ribosomes work together to ensure accurate information transfer using base pairing rules. This covers the detailed process of the central dogma. I hope this helps you understand how life executes its genetic instructions.